So as you are probably quite familiar with, well, I wasn't going to introduce the PRRS virus because you guys are all familiar with it. And as you are also probably familiar with how the PRRS virus enters, I'll just briefly cover that. The theory is that the virus comes in and has a loose association with heparin sulfate. Then it binds to the N-terminal region of cyclohexin. That gets internalized into a vesicle. The vesicle then, the internal pH drops. CD163 then associates with the virus, unpacks it so the genome can actually enter the cell and infect it. And if we look at Van Breeden had a real nice diagram, and you can see on here where the virus comes in at the top, associates with heparin sulfate, binds to cyclohexin, gets internalized, pH drops, associates with CD163, and the viral genome is uncoded and released into the cytosol so that it can infect the cell. There's a lot of evidence, and I'm going to go through some of the evidence here. When we first started on this, we thought, yeah, cyclohexin is a really good candidate. So Dwan generated alveolar macrophage-specific antibodies, and two of these antibodies recognized a 210,000 molecular weight protein. The protein was purified, identified as cyclohexin. And if we took that protein and re-expressed it in non-permissive cells, you could get virus binding and actually get the virus to be internalized via endocytosis. And the molecule, the cyclohexin, actually co-localized underneath the plasma membrane with the PRRS virus. And there was some sialic acid binding sites mapped to the N-terminal region, and in fact, if you deleted the N-terminal region, you could delete that binding. In fact, it was narrowed down to amino acid 116, whereby there were some studies that suggested if you did an arginine to glutamic acid mutation there, you could abolish binding. And also interestingly, pig, human, mouse, and all cyclohexin all permit entry of the virus into the cells. So we thought, we can knock this out. So we tried to knock out exons 2 and 3, which represents amino acids 17 to 235 plus. Incidentally, the signal peptide is amino acid 1 to 57, and we hope that this would reduce the binding of the virus to the cell, thereby reducing the infectivity of the virus. So this is a cartoon showing the genome, and up here at the top we have the exons labeled in blue. We designed a targeting vector that had 3 bases out of exon 1, exon 2 and 3 eliminated and replaced with the neomycin-resistant gene, and then exons 4, 5, and 6. And so if we actually had our integration occur, we would come down the chromosome, we'd come into exon 1, and then we'd have a few stop codons along with the neomycin-resistant gene, and we would hopefully have animals that would not get PRRS. So we did identify a targeted clone. We used homologous recombination, and it was a little bit of a challenge. We screened over 2,000 colonies, and if you look at the efficiency of this, it's not very efficient, 7 times 10 to the 6th. Then we did somatic cell nuclear transfer, and this involves taking an unfertilized egg up here at the top, removing the metaphase chromosomes along with the polar body, then taking our donor cells, which we've genetically engineered, and maybe we've done other things to them. We then transfer that cell into the space in the zona pellucida along with the unfertilized egg. We then fuse the 2 cells together. That deposits the nucleus into the cytoplasm. Hopefully it gets reprogrammed, remodeled, and we activate the egg, and hopefully that will develop to term. Here's kind of what it looks like. This is about 150 microns in diameter. The unfertilized egg, there's the polar body. We treat these cells with cytoskeletal inhibitors so we can actually push the pipette through the zona pellucida and into the cell, but we never penetrate the plasma membrane. It's like sticking your hand into a balloon. You pull out the pipette, the membrane pinches off, you can spit the contents back out of the pipette, and here you have your enucleated oocyte. We can then go pick up our donor cell, which incidentally is out of focus compared to the egg because it's on the bottom of the dish, pick it up into the pipette, stick it back in the same hole, and now we have our nuclear transfer oocyte, but we still need to fuse these together, so here's a potential litter of clones. We place them between a couple of electrodes, one at the top and one at the bottom, and we place them so that the electric pulse will go through the donor cell and into the oocyte, basically so the plane of fusion is parallel to the electrodes, and that serves two purposes. That fuses the two cells, gets the nucleus in, and activates the egg so that it begins development. We did 2 embryo transfers, and you can see this was done back in 2010, and we had 6 pigs born from one transfer and 2 pigs delivered by cesarean from the other. Well, this is what they look like, but remember they're all heterozygous. So we don't really have an animal that has no siloadhesin yet. These pigs still have one good copy. So our founder males were mated to wild-type females, and that gave us an F1 population, so that now we had both boys and girls that were heterozygous that we could then mate together to produce homozygous offspring. And as you recall, when you do that mating, you've got animals that have one good copy and one mutated copy, males with one wild-type copy and one mutated copy. A quarter of the animals that you produce should be homozygous knockout, a quarter should be wild-type, and half of them will be heterozygotes. And this was our first pig born, or our first litter born. This was born in October at 12. We had 4 that turned out to be homozygous knockout, 4 that were heterozygous, and 4 that were wild-type. So it worked really well for doing the challenge experiment. We shipped these pigs to Kansas State University. They were acclimated and then challenged with KSO6. The blood was sampled days 0 through 35. They were assessed daily for clinical signs. They were co-housed, so we had controls and mutated animals together. There was no apparent clinical sign before infection. After 3 days, they developed exactly what you would expect, and most of that resolved within 1 to 2 weeks. At the end of the study, they were euthanized, and histology was performed, and there was really pretty much no difference. All the pigs have moderate to severe multifocal interstitial pneumonia. We also collected alveolar macrophages. So Dr. Rowland's group did flow cytometry, and this is what you would expect with wild-type pig, where we have our siloadhesin peak, and then our CD163 peak, and our secondary antibody. So on a wild-type pig, that's what you would expect. With our homozygous knockout pig, so here's our wild-type pig again, here's our knockout pig, and you can see that the siloadhesin peak is shifted over to be not different from the secondary antibody, implying there's no siloadhesin on the porcine alveolar macrophages, whereas we still have our positive control, our CD163. And if we look at all the pigs, here's the wild-type pigs. They both showed 2 nice peaks. The heterozygous pigs, we had 3 of them left. One of them didn't make it through the study. We have, again, 2 nice peaks. And with our knockout pigs, all 4 of those, again, there's no peak for the siloadhesin. It's not different from the secondary antibody control. So if we look at virus isolation and look at days after infection, we've got our 3 different treatments, our 3 different genotypes, and there's really no difference, unfortunately. If we look at our viral RNA, again, pretty much no difference between the 3 genotypes, days after infection. If we look at IgM, you might convince yourself there's a little bit lower here, but our numbers are small and there's a lot of variation, so again, we pretty much conclude there was no difference in IgM production. If we look at IgG, you know, the knockouts and the heterozygotes might be a little bit lower, but again, our sample size is pretty small. Nevertheless, the pig still had symptoms of PRRS infection. So we conceived this idea back in 2002, and it took a long time to get money and get people convinced that we should be able to do this, and we kind of played with it throughout the period. Then we got serious about 2009 and really pushed forward on this and produced the founder males January 11, and publication came out last year, 2013. 11 years from the time we had the idea to we actually had the answer. Now, that didn't give us the answer we want, but part 2 is, and part 2 goes a lot faster. Well, first of all, siloadhesin, it's not embryonic lethal. We know that the knockout mice have a subtle changes in T and B cell populations, reduced IgM. Maybe we saw some of that. The siloadhesin knockout pigs may have an altered IgG response, but there was pretty high variation. Sample size was small, and they didn't affect susceptibility. We have transcriptionally profiled the alveolar macrophages, and we're working on that manuscript right now. Incidentally, if anybody wants cells from these pigs for any diagnostics or their use, they're going real cheap right now. Okay, so the case for CD163. I'm not going to go into the details on it, but there's a whole series of experiments that were done similar to what I described for siloadhesin that provide evidence that CD163 is involved. And Welsh and Calvert did a really nice review of this a few years back. If you want more details, I encourage you to go read that review. CD163 is a member of the scavenger receptor cysteine-rich superfamily. And what we're really interested in is right here, this exon 7, or exon 7 encoding domain 5. So the numbering kind of gets a little bit confusing. But what I wanted to do was focus in on this paper by Van Gorp, where they tried to determine which domains of that CD163 were really responsible for infectivity. And so they took cells and they transfected them with both siloadhesin and different CD163 constructs. They inoculated them with the PRRS virus, and then they counted infected cells. And so CD163 is a member of a gene family, so we have CD163 here with domains 1 through 9. And here we have, actually this is going to be human, and here we have human CD163-like. And so they took advantage of the human system and the mouse or the pig system and did some swaps. And what they came up with is a system where if they had pig CD163, there was infectivity. Human CD163, there was infectivity. Human CD163-like, it dropped down to nothing. When they swapped out 7, 8, and 9 in pig CD163 with human CD163-like, they abolished reactivity or infection, and they narrowed it down to this domain 8 that they could swap out with domain 5, and they ended up with no infectivity. So we proposed a couple different ways to do this. One was, well, let's just go in and do homologous recombination and make that same swap so that we have human CD163-like domain 8 and stick it into pig CD163 domain 5 and get rid of domain 5. So that was our goal when we first started this. And we thought that that was really important because domain 3 is involved with haptin hemoglobin metabolism. Domain 2 is involved with erythroblasts. Tweak receptor is in 1 and 4 and maybe 6 through 9. Bacteria are cleared by domain 2. African swine fever virus, it might be in there too. And PRRS, well, we think that's domain 5. So, but is a domain swap necessary? Haptoglobin's ability to promote binding of hemoglobin to CD163 in humans has high affinity, but not in mice. In mice, CD163 accounts for a small part of hemoglobin clearance. So in humans, you're going to think it's probably more important. And in fact, knockout of CD163 results in, and I quote, no apparent phenotype change, and offspring were viable and fertile, and that's in mice. So we move forward to try to genetically modify the CD163 by transfection, and we made this domain swap targeting vector to knock out CD1, and we also wanted to knock out CD163 with non-homologous injoining. We also wanted to replace that domain 5, and then I'm going to talk about it using CRISPRs here a little bit by injecting those into zygotes and mutating CD163. So I don't really want to go through all the details on this, but we worked quite a bit, and we performed 38 transfections. We screened 3,399 colonies with our domain swab using traditional homologous recombination, and we had nothing to show for it. And it was really quite disappointing, and we really needed some help, and that's about the time people started talking about using these CRISPRs. And so what are CRISPRs? CRISPRs, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Basically, this is a system that bacteria use to destroy invading viruses. And they take a piece of that genome and they insert it into their system and cuts it up. So people thought, well, maybe we can do that in pigs or in mammalian cells and use that as a method for genetically engineering. So DeMaio and Spencer talked about CRISPR bacon, and yeah, that's what we're going to talk about today. So you can use this Cas9 CRISPR system to, along with a guide RNA, to cut your genomic DNA. It induces double-strand breaks, and you can either have those repaired by non-homologous injoining, where you may get a single base pair deletion, insertion, inversion, indel. You can get all kinds of things. Or you can potentially add an oligo or a targeting vector, and hopefully you'll get homologous recombination and insert the piece of DNA that you would like to insert. So not only can we use this on somatic cells, we can also inject just the CRISPRs into zygotes, and we did that, and I'll describe some of those results here in a little bit. So when we did somatic cell nuclear transfer with our CD163 mutated cells, we did 7 embryo transfers. We produced 6 pregnancies. We had 32 piglets. 2 of those were wild type. Now I say wild type cells. They started out wild type. We tried to mutate CD163 in it. We also did 6 embryo transfers where we started out with siloadhesin knockout cells, and we tried to introduce the CD163 mutations. 2 of those maintained pregnancies, and we had 5 piglets from that. We also did zygote injection. We did 2 embryo transfers. 1 maintained the pregnancy, and we had 4 live pigs, and all 4 of them had mutations. This is a list of the mutations, and I do not expect you to read it or get anything out of it other than, wow, what a mess to sort through. Because we had all kinds of different mutations. Or you could say, wow, what a mess we have to sort through. This is good. Because you can translate that information and come up with predictions on what the protein would look like. So here's some of our piglets. This is after somatic cell nuclear transfer. This is litter 63. This is a wild type band. All 3 of these piglets have deletions. Here's litter 64. Here's the sow with another wild type band, and all 4 of those piglets had deletions. When we look at the zygote injected piglets, we had a number of different mutations that occur. Here's in number 1 we had a deletion. In number 2 we had 2 sizes of deletions. In number 3 we had a mutation of an A, single base pair. In this piglet we had 130 base pair deletion and 132 base pair deletion. So when you translate those different genotypes into protein, well, what do you get? Well, here we have our human CD163-like. Here we have human CD163, and I see my numbers, everything didn't come off quite right. Nevertheless, here's our wild-type pig. Domains 1 through 6 are present. Here, oh, good, that one, we don't have domain 5. That's interesting. Look at this one. We don't have domain 5 or domain 6. This one, we have a partial deletion of domain 5. This one, we have a partial domain swap of domain 8. And I should emphasize that we didn't use a human gene. We introduced the mutations into, we just basically synthesized an oligo with those 33 bases changed that are different between pig and human in that domain. And so we didn't really use human DNA, so don't worry if you're going to eat it, it's not going to be human. You're supposed to laugh when I said that. Okay, so the question is, can PAMs from these pigs, mutant pigs, be infected with PRRS? 8 of the pigs we had ended up euthanizing because we really didn't have space and we didn't have an opportunity to go into the system at Kansas State to challenge the pigs. So we went ahead and euthanized the pigs, collected PAMs. We shipped them to Kansas State, and while they were there, they did some things like looking at flow cytometry. And this is CD163, and here's our wild type, and here's our CD163 knockout pig. It's like the protein's gone. This is good. And then they also took some and infected them with PRRS for 24 hours and stained them with an anti-PRRS antibody, and then they measured infection by antibody presence. And here's our genotypes, and of the 4 that we sent, here's what we came up with. The wild type, there was infection there. The partial domain deletion, there was infection there, but the deletion of domain 5 and the deletion of domain 5 and 6, they didn't see any infection. If you want any details on this, please don't ask me. Please ask Bob Rowland. Okay, so overall conclusions. We produced pigs, siloadhesin pigs, and they are not resistant to PRRS. Knockout of loss of function mutations of CD163 pigs are completed. There is some, at least in vitro, resistance to PRRS infection. We also have animals that have siloadhesin knocked out and CD163 knocked out. So will it prevent infectivity? The challenge studies will be done in the spring and summer of 2015. The knockout and domain 5 and 6 deletion pigs are confirmed to be pregnant, so we're awaiting their delivery and shipment to Kansas State. And hopefully this will enable us to identify what are the molecules involved with entry of PRRS virus into cells resulting in infectivity. You can identify all the different molecules you want, but until you go in and knock it out and don't get infectivity, you don't know if you've got the right molecule. So we come back to the model. I think we've crossed siloadhesin, at least by itself, off the list as a entry mediator. Something else is certainly able to carry it in and permit infectivity. But hopefully this is a model. This is good. This is a model that you can build testable hypotheses based upon, and you can answer the questions. So the timeline for the CD163, I told you the siloadhesin took a little while. Well, here's the dates. We started in July of 13, and we had our piglets by January of 14. It only took us 5.5, 6 months to get the founder pigs. It's a little shorter than 11 years where we started. These CRISPRs have just changed things completely.